Καλησπέρα.
Είναι μεγάλη μου χαρά σήμερα να παρουσιάσω τον Γιάννη Τσιβερδή. Ο Γιάννης είναι νευρολόγος, έχει τελειώσει την ειδικότητά του στην νευρολογική κλινική του Πανεπιστημιακού Νοσοκομείου Ηρακλείου. Έχει αποκτήσει μέσα στα χρόνια ένα ενδιαφέρον για την νευρογενετική. Μάλιστα το διδακτορικό του είναι, στο εργαστήριό μας είναι πάνω σε αυτό το θέμα, στις νευρογενετικές παθήσεις. Και σήμερα θα μας μιλήσει για το ένα ευρύ θέμα, που είναι η γενετική στις άνοιες, ξεκινώντας από τα πιο απλά και πηγαίνοντας έτσι στα τα πιο σπάνια και τα ίσως πιο απλά, στα πιο σύνθετα και τα πιο συχνά. Γιάννη, ευχαριστούμε πάρα πολύ για την αποδοχή της πρόσκλησης και περιμένουμε να σε ακούσουμε.
Γεια σας και από μένα. Είχα την εντύπωση ότι θα πρέπει να κάνουμε την παρουσίαση στα αγγλικά.
Ναι. That's correct. Αυτό είναι σωστό. Παρότι περισσότεροι συμμετέχοντες είμαστε στα ελληνικά, νομίζω καλύτερα στα αγγλικά. Θα αρχίσω ξανά. I'm very happy to introduce IoannisTsiveridis. Ioannis is a neurologist. He finished his residency in the University Hospital of Crete in the neurology department, and he has been increasingly interested in neurogenetics. This is the topic of his PhD dissertation that he is currently pursuing in the neurogenetics laboratory in the University of Crete. And he's going to talk today about the genetics of dementia, starting from the rare diseases and going on to the more common ones. Gianni, se acum. We are eager to hear you.
Hello. Hi to everyone. Thank you for the invitation. I'm very happy to be here. So I will go to screen share to start the presentation. So our topic today is about genetic of dementia. We will start talking about rare disorder. Slowly we'll go to more common dementia types. So there can be one mutation in a specific gene, a single gene disease, or can be a complex disease with multiple variants in different genes. And in this complex disease, there is the environment plays an important role. In the past, we have some gene testing that was targeting specific mutations, like Sanger sequencing. But in recent years, we have developed new sequencing techniques like whole exome sequencing and whole genome sequencing that can test thousands of genes at the same times. And this is a technique that we use also in our laboratory. My speech today will be about a top part of my thesis, study of neurogenetic disease in Crete, and is a presentation about different studies that we have done here in our laboratory. Not only mine, especially, but from other patients also. So we will start from the most rare types of dementia, and we slowly will go to more common. First is Lafora disease, quite a rare disease. It's about an 80-year-old woman that from a young age, 3-4 years old, she was starting to have a myoclonus. She was non-diagnosed until she came to us, but from 14 years old, started to have a problem with vision. She started to have a gradual decline in cognitive function. And when she came to us, we said that she has no family history. All the examinations were normal, even the MRI was normal. The girl who was having seizures and he was in medication with levetiracetam, and she was continued to have seizures. We step up the medication with valproic acid, but the girl continued to have generalized seizures. And then we are in dead end in perspective of what is the diagnosis, and we continue with whole exome sequencing. Now there is different causes that can produce progressive myoclonic epilepsy. There can be LAFORA, like in mutation EPM2A gene, or can be something like mitochondrial disorders like MERV syndrome, and other causes. But here in our test with whole exome sequencing, so that this woman had a mutation and a variant in the EPM2A gene, and this is a mutation that is in the part in the exon 3, in the part of the gene that is we have found until now 4 families that have this mutation. Actually, this is the we have found that has this mutation, that this mutation, the specific mutation, is 4 families that have that is known to have a LAFORA disease. So it is a known mutation. And because we have these findings, we want to investigate it a bit further. So we did an axillary biopsy, and we identified that it has a LAFORA bodies that's characteristic in this disease. So the diagnosis here is LAFORA disease. It's autosomal recessive genetic disorder. There is a disorder in the metabolism of glycogen, and this person have a myoclonus epilepsy, like this woman, dementia, and the characteristic, as we said, is the LAFORA bodies that we found in biopsy. And this patient is published in this recent paper FAS that has also another a few other patients with genetic epilepsy. Continuing to something more common but still rare is the CADASIL. CADASIL is a disease, a vascular dementia, genetic vascular dementia. And here is we started with a 67-year-old woman who was having a headache from a young age. She had a depression. She was suffering from 4 strokes. After that, she was starting to have dementia. And we did a MRI, brain MRI, and we see that she has a diffuse leukoencephalopathy, and actually, as you see here, has affected a lot the temporal lobe. The temporal lobe is a characteristic place that affects the CADASIL. And so we continue with these findings to whole exome sequencing, and we identified 2 heterogeneous changes in NOTCH3 gene, as you see here, in cysteine 206 tryptophan, and also in valine 2021 methionine. And we thought that this is a case of CADASIL and genetic vascular dementia. But this woman has also a son, a 30-year-old son, that he had a headache from a young age also, like his mother, and he has stress disorder and sleep difficulties. And because we knew his family history, we pursued with MRI also, and we found leukoencephalopathy, not so diffuse like his mother, but there is also some findings also in temporal lobe. So we continue with to try to search about any variant in NOT3 gene, and actually we identify the variant in cysteine 206 tryptophan, the same that his mother has. So this is the pedigree of the family. As you see here, the mother and the son, and you see that they have the same variant. And this is we know that patients that have a change in NOT3 gene that are a patient with CADASIL, the mutation, the majority of them are in exon 4, and our variant that we speak is in exon 4, and we know that this is a change in epidermal growth factor-like region that plays an important role in this disease. So this is actually this variant is new. It's novel, the variants, and the first one, the Val in the 2021 methionine is probably benign, and that's because there is a conflict data. We know that the frequency of this variant in the population is actually quite larger than what we expect in this disease, and we have detected this variant in healthy controls, but the predictive computing software showed us that this is a harmful variant. So the data here is conflicting. We're not sure. We don't believe that this is a pathogenic variant, but the other variant, the cysteine 206 tryptophan, it's probably a pathogenic change, and that's because there is something based on the American genetic medicine criteria, that first, there is no mention of this variant in the population database that are known. Second, the place that there is this variant happened is in the place that there is a region that's functional for the gene, and in this region, we don't find any benign change. And except for that, we know that the difference, the physicochemical difference between cysteine and tryptophan, it's quite important. And also we have the computer predictor software, so a harmful that was a harmful variant with a CAD over 15. And taking into account that not only this criteria, but also we have the family that has the same variant in the mother and the son, we believe that this is a mutation, probably pathogenic change. And this actually it's a novel and a published data. We hope to publish in the next weeks or months. Continue to something more common that we see more often is frontotemporal dementia. And frontotemporal degeneration is it's something that is quite interesting. There is a clinical, pathogenic, and genetic spectrum that's very wide. And what this means is that in clinical perspective, we see that it can be from ALS to frontotemporal dementia to cerebral to progressive dementia or other type of dementia as you see here, like contravascular degeneration. And except from the different disease that we see here, we see different pathologies. It can be a DDP43 pathology, or a tau pathology, or a fuse pathology. And there are different genes that can cause this kind of diseases. In the DDP43 pathology, we see that can be a change in the GRN gene, or can be an expand repeat in C9ORF72 gene, or it can be in VCP gene, TRP DP gene, or TPK1 gene. Or in the tau pathologies, we can see that can be in a variant or mutation in the MAPT gene. So we did a study in this population. Actually, we take a population of 130 patients with frontotemporal dementia, ALS, or a combination of those two. And the demographics here, the patients have, there was no difference in sex, in age, or in level of education. We noticed that the frontotemporal dementia is starting earlier, or some things that we know. We also see that the ALS has a shorter duration of disease. That's something that also know. And also we see the mental test and FAB score were lower in the patients that had frontotemporal dementia, or frontotemporal dementia with combination of ALS. The method that we used, we tested for biomarkers with ELISA in CSF, and we checked for beta amyloid, for total tau, or for phospho-tau 181, or we check for DDP43. We also check separately for repeats in C9ORF72, and we use also whole exome sequencing. And as you see here, how would we divide and we address this patient? First, from this 130, we did to 14 of them whole exome sequencing right away, or for one of them target gene sequencing, because they have a specific clinical characteristic that suggests only one disease. And from the rest, 115, we check for the C9ORF72 repeat expansion. We found 15 patients that have this repeat expansion, and from the rest 101 that didn't have the repeat expansion. From those, we did whole exome sequencing to 81. As you see here, we found that from all those patients, the 56 had a frontotemporal dementia, and from those, 5 have a repeat expansion in C9ORF72 repeats, and we find that there was also a progranulin mutation in 3 of in the GRN gene. We found variants in 3 with VCP gene. And with those that we have ALS, it was 58 persons, and from those, 6 have repeat expansion in C9ORF72, 2 have a mutation in TRDPP, and 1 in fuse and 1 in SOD1. And lastly, the smaller group was the combination of frontotemporal dementia and ALS, and there was 16 patients, and from those, the 3 had a repeat expansion with C9ORF72, and the 1 had a variant in TRDDP. Now, here you see the 14 persons that have a repeat expansion, and you can find all the details about all these patients in our published data, as you see here. And here is the patient that have no repeat expansion. And the important thing is to see here the genes that we speak about. TAR, DBP, GRN, VCP, FUSE, and SOD1. And when you see those patients, the group that has a repeat expansion in C9orf72, we see that has a younger age of disease onset, and also they were 10 years younger when they entered the study. And when we try to see those patients about the biomarkers, we found that the patients that have this repeat expansion have a lower level of phospho-tau 181 levels in the CSF in comparison with those that didn't have the repeat expansion. Also, we wanted to test the plasma, and we test the plasma from progranulin that's suggestive, or that's pathogenic that's suggesting a pathogenic variant GRN, and we see that also the plasma was lower in those patients. That's confirmed that that's something that we expect. We expect in these patients that have a pathogenic variant with GRN to have a lower plasma level on progranulin. And this is also the published data, the publication that we did, and we have all this data there. And the last one is the Alzheimer's disease. Is the most common cause of dementia. There's a lot of numerous genes that can cause dementia, but the most important is Breslin-1, 2, APA gene, and also the ApoE alleles that affect this disease a lot. And we did a study here that we use 4 different cohorts. The first was the Crete aging cohort that we have here in Crete. The other one is the SCEPSI program that has a lot of patients with mental problems, impairment in function, and is also in Crete. And also we have the Alzheimer's disease neuroimaging initiative. This is a cohort that is worldwide, and actually it's a cohort that is not Cretan population. It's not from Crete population. It's worldwide without Cretan here. And last, we check also the cohort of in Crete that we have with frontotemporal and ALS, 10 patients that have this disease. Now, the Crete agent cohort that we speak about, this is the region that the families are from, Heraklion. And how we approach this cohort, we start to investigate 3,000 participants that were older 60 years old, and we screened them with mini-mental. The population that has mini-mental lower than 24, we thought that is mental impairment in those patients, and so from those 344 accepted to further investigate it, and from those we diagnosed in 109 dementia and 159 mild cognitive impairment. And for those that they have dementia, we succeed to do a whole exome sequencing to 95, and those who have mild cognitive impairment, we did whole exome sequencing to 20. From the patient that was over 24 mini meltdown, we find 161 that accept to further be further investigated and to be in our study. Of those 161, the 112 diagnosed as non-impaired cognitively, and we did a whole exome sequencing to 81 of them. Now, to all those patients, we take blood to analyze the DNA. We did a whole exome sequencing. We used the Ingenuity program to analyze the data, and we check for specific genes. We check for the 4 genes that are most important for Alzheimer's disease, like AP, Breslin 1, 2, and APO. We check also for 14 genes for frontotemporal dementia, and also we check for GLUT1 and GLUT2 genes. That's 2 genes that we are very interested in our laboratory, and there is literature suggesting that they have a close relationship to neurodegeneration. And what we found, the genes that we found, we confirmed them with singular sequencing, and for the APO, we confirmed them with PCR-RF-LP. Here you see the GLUT2 gene and the protein that they produce, and actually we did a crystal structure analysis here. Also, I want to say that in the program Skepsis, we use a different method to approach it. We take also blood from DNA, but from DNA test, but we take 235 patients, and from those we did the CASP method that's more specific. And also we examined them neurological, psychiatrically, and we did numerous neuropsychological tests. And the results in the Crete aging cohort, we found when we did the whole exome sequencing, we find all these, we investigated for all these genes. We found that the GLUT2 gene, the one that we said that we are more interested in, that has a role in neurodegeneration, we found that serine 498 alanine is more common in controls, and this is statistical significant. And also we find that in gene TAR-DPP, we find that isoleucine 383 valine had this variant in 2 patients with Alzheimer's disease. And why this is important? The mutation in the TAR-DPP gene is mutation that we see in patients that have frontotemporal dementia and not Alzheimer's disease. But here we find 2 patients with Alzheimer's disease that have this variant. And because we want to investigate this a little more, we try to check in different cohorts that the Crete aging cohort, and we check the GLUT2 serine 498 alanine. We check it in the replication Crete cohort that has double numbers of participants, 400, and we also investigate in the ADNI cohort, that's the worldwide cohort with 800 participants. And to investigate the mutation, the TAR-DPP gene, we investigated in the cohort that we have with frontotemporal ALS cohort with the 10 patients. And we will see further the results. So we did a principal computing analysis for those for those cohorts, and what we see in comparison with the 1000 gene project, as you see, it's this green. We see that the black one, that's the Crete aging cohort, we see the black one is this, and we see that it is actually very homogeneous and not spread. And also it's very similar to Tascani group in Italian group of persons. And that's suggesting to us that it's a group that's quite homogeneous, and we can have more solid results for this. And as you see here, the demographics, you see the this is the three groups: Alzheimer's disease, mild cognitive impairment, and controlled. And you see that there's a bit similar, but there is a bit older in Alzheimer's disease. We see that is more women in the Alzheimer's disease, and this is something that we expect actually. It's more common as Alzheimer's disease in women. And if we go further, we see that in Alzheimer's disease group, there was a higher percentage of family history for dementia, and also the mini mental score was lower. An important thing to note here is the APO E4-ALI is actually 3 times more in Alzheimer's disease patients and mild cognitive impairment patients. And this is something that is actually goes with what we know so far in literature. Here is the results, as you see, with the genes, and from all these genes that we identify, one important thing, the two important things that we speak a bit earlier is from Cloud2 gene, that there is the serine 498 alanine, and you see there is 2 patients in group of Alzheimer's disease, 3 on mild cognitive impairment, and 13 in control. And this is a big difference and statistically significant. And also for the TARTPP gene, we see that the isoleucine 383 valine is there is 2 patients in the group of Alzheimer's disease, as we said earlier. And the comparison of those data with the other cohort, with the cohort of Skepsis and the ADNI, we see there is the pattern is similar. As you see here, that you will see that there is also in the this is the finding for the CLUD2 gene. Serine 498 al, and you see that is also in the other group a bit higher. In the group of Alzheimer's disease and mild cognitive impairment is 5%, and in the control is 8. In the ADNI cohort is 3, and in the control is 3.2. It's not a statistical significant this change, but this difference, but it shows us a pattern. In summary, this study shows us that first, it's actually quite important, is that this variant in isoleucine 383 valine is that usually we see in frontotemporal dementia is in 2 patients of our cohort with Alzheimer's disease, and that's something that's actually quite interesting. And the other thing is the fact that we find the serine 498 alanine more in the control group, give us and make us think that probably this is has a protective role for dementia. And because we wanted to investigate this a little better, we go to the Cretan bank, brain bank that we have, and we do immunochemistry in beta amyloid and hyperphosphorylated tau protein. And when 10 patients that have that was diagnosed and confirmed that have Alzheimer's disease, and from those we did immunohistochemistry for TDP43 protein, and we found in 9 of those 10 that have an inclusion immunocreatant to TDP43 protein, and that's something that also suggests the findings of the previous study, that this protein and this gene plays a role not only in the frontotemporal dementia, but also in Alzheimer's disease. And this is the publication that we did recently, showing all these findings. In conclusion, I want just to say that Alzheimer's disease, frontotemporal dementia, and the other rare causes of dementia have actually an important genetic basis, and we need to be aware of it. This kind of disease have a high phenotypic genetic heterogeneity, as you see, and it's actually quite difficult for us to make a diagnosis. And now the new generation sequencing that we have and you use, it's actually more very accurate in comparison with the previous that we have and the other tests that we did in the past, and can help us to give a genetic diagnosis and to help those patients and their families. And hopefully with the revolution in the genetic treatment and medicine that we have now, we will be able in the future to give also a treatment to those patients. Thank you. This is the reference. It's a bit small, but thank you very much.
So, Yanni, thank you very much for this presentation. It was a journey from rare diseases to more common ones. Of course, as you said, it's very common for dementia to have a genetic basis, but also you have the, as you mentioned in the beginning, you need the environment to exert its effect on the genetic background of the individual. So the lecture is open to questions. We will be happy to, I'm sure Ioannis will be happy to discuss these findings.
Yanni, may I ask something?
Of course.
Yeah, okay. Hi. First of all, congratulations to you and the whole group, for the work you're doing and for the presentation. The Alzheimer's cases where you identified the FTD mutation, were they any different clinically? I'm sorry, because some of the questions may not be very valid because I had some connection problems and I was in and out, I'm on the road, but were they clinically any different? Phenotypically, I mean.
Phenotypically, no, actually. They have the typical presentation of Alzheimer's disorder on how they started. They didn't have any behavioral or speech problem that suggesting the frontotemporal dementia.
Okay.
But
were they younger? Were they younger, or so they were like older amnestic people, or?
No, they was actually they were older. I don't remember the exact age, but they were older. The thing is that actually we don't have verified Alzheimer's disease with biomarkers in those two patients, but we have the clinical imaging test that suggests.
Right. And in the literature, how common is that? I mean, are there previous studies reporting these mutations with an Alzheimer's type phenotype?
It's actually something that usually you don't see. It's there's not a literature data that have those findings, and I think that's one of the reasons that we will be able to publish this.
Okay, and one last thing, not to monopolize the question, is from the Cretan aging cohort, how did you select those who would undergo whole exome sequencing from normal, from MCI, from demented? It was first come, first served, or it was some peculiarity in their presentation, or what was the criteria?
Actually, the criteria was first to have a minimal under 24. I mean about the people that we did whole exome sequencing that we said that have dementia or mild cognitive impairment. The thing that we wanted, it was not to have other diseases, and not to have a stroke or things that can affect the results. The other thing that is, I think, important is that For those patients, as you see, we start with 600. The 344 accepted, but we did whole exome sequencing to around 100 with Alzheimer's disease and mild cognitive impairment, and that's why a lot of them, when we further investigated, we see that the low mini-mental was not it doesn't show actually the cognitive impairment. There were some have depression, some have anxiety, and that's why the lower values on the test. So we filter it clinically with medical history, actually.
Hold on, I'm a little bit confused now. I'm actually more confused. So from the 600, what do you mean 300 accepted? Accepted what?
Accepted to do gene sequencing.
Okay. So the others, so you had blood draw also, you mean you had blood specimens for the others, but you asked for explicit consent for this type of analysis?
I mean, in order to participate in the study, this cohort of Crete aging cohort is known. We filter it with mini-mental test, and after this filtering with a mini-mental test, we try to take this patient to put it in our study. That's a different study.
So hold on, wait. So there's a separate study that is
let me say it a little simpler.
Yeah.
We have the Crete aging cohort, that's 6,000 people. Okay. And we filter those patients all with mini-mental. And the mini-mental show us which of those will go to which category, the category of over 24 or lower of 24. Those patients, we wanted to further investigate to see what this low mini-mental or high mini-mental actually mean. But in order to do this, we need to have the patient agree to do some test, mental test, to give us a family history, and after that they need to give us blood and to give us the permission to check the DNA. So in the bottom line, from 600 that have the low mini-mental, for example, we have 100 that accept the whole procedure.
Okay.
Some of them, it's not the people that didn't accept it. Some of them actually doesn't, for example, have lower mini-mental score, but didn't have actual dementia, were depressed. Those wasn't in the study.
If I can add, as Ioannis said,
maybe I can help. The reason I'm asking is even from those who agreed for a more extensive evaluation, even from them, it's a subsample. So whether there's any additional filtering in order to give us a sense of how representative is that of the dementia population and have a sense of the frequencies. That's what I'm asking.
Yes, as Ioannis said, from dementia, if I may add something, 109 persons were diagnosed with dementia. And this, from this, we selected 95 that were fulfilling the criteria for Alzheimer's disease. The reason we excluded the other 14 patients were either what Ioannis said, that maybe there was coexistent depression or other problems, or they didn't have Alzheimer's. So we didn't want to, let's say, have an admixture of frontotemporal dementia, Lewy body dementia, vascular dementia. So as Ioannis said before, if a patient had many strokes in the past, we didn't want to have like...
that's clear. And the other ones, the MCI and the normals?
The MCI, we selected only 20 of these, and the normals were 81. The reason, in addition to budget availability, was that we tried to have like a matching for place of residence. We couldn't match because of the small group size for everything, but we thought that being a resident of the same village, a dementia, an Alzheimer patient, and the control means that somehow they share a lot of similar genes. So our main matching criterion was the place of residence.
Okay.
And actually, somehow we were, let's say, somehow happy to see that, as Ioannis showed in the principal component analysis, our sample was homogeneous. And this is important. You saw in the picture that in the diagram that Ioannis showed that compared to 1,000 population study, it's concentrated in a specific area. So this gives, we think, an advantage compared to studies from other places where you have like people from different, genetic backgrounds. So if you have a homogeneous population, maybe you have a better chance to have, let's say, a signal in some of the genes.
Okay, thank you.
We thank you for the questions. And sorry.
It's okay.
So is there any other question? I think Dr. Liketsos wants to ask something.
I do. Very, very nice work, and thank you for presenting it. It's a complicated set of issues, but really I was going to raise a question about possible future methodologies. So you've identified a few rare variants. I'm wondering whether you have available PBMCs from these individuals. Because if you do, it offers you an opportunity to make stem cells out of those, create cortical neurons, and compare functionality to see if you can detect the effect of the variants in individuals, which would be really very interesting.
So can I intervene on this? I'm very happy that you bring this up, because I have been begging Ioannis Haralabopoulos since several months to take this TDP43 mutants and produce IPS from this. Yanni, you are still here?
Yes, Dr. Liketsos, actually this is first in line. I hope soon enough we'll have some news from there too.
That's what he says to me since about 1 year.
We have to put the basic first, Ioannis, and then do the right work.
I'm joking, but it's a very nice question.
This is actually very interesting, yes.
Yeah, let's follow on the theory. So Yanni, Yanni, I speak to both, all three of you. Gosh, this is a challenge. So Yanni, all of you, what are you going to do with the stem cells? What are you thinking you will study? You'll differentiate them into some form of neurons and neuronal precursors, and then you'll assess some aspect of their functionality. So I'm just curious what the plans are.
Yanni, do you want to
No, you are more,
What I had in mind first of all is to see the differentiation process compared with a control, let's say, cell line. So to see the differentiation process towards neural stem cells, first of all, and then to mature cortical neurons. Then the other thing that we have established the last year in the lab is to specifically differentiate these cells into oligodendrocyte cells. So we could examine then their ability to myelinate or in co-cultures with neurons and see if there is any deficit there also, and things like that. And if you ask my personal interest, then, and we work on this actually, that's one of the reasons that we delay a little bit Ioannis' ideas, is to see any difference in the effects of growth factors, specific growth factors, and the receptor towards differentiation of the cells. So this is some very preliminary questions that we have for this, for AD-related cell lines, let's say.
And what were you thinking would be the controls? You would have other AD who are not TDP variants and some normal older people who have nothing. Yeah.
That's the other thing actually that we were discussing just yesterday with Ioannis, that the proper control should be actually two. One AD that does not have this specific mutation, TDP43 or ApoE4 or whatever, but they still are coming from AD patients. And the absolute control would be similar age, healthy donors to see all these three conditions compared to each other. That's absolutely right what you said. Yes.
So if I may add, I mean, we have been looking to A beta peptide and to tau all these years. I think there's something more in there. So that's why, as IoannisTsiveridis presented, we were very interested to see that on one hand, you have like variants of TDP43 that are causing frontotemporal dementia, are causing ALS, are causing both, and are causing an Alzheimer's disease phenotype. So, and on the other hand, brains from our brain bank that we have presented in a previous HIAAD lecture that are well characterized, and we find the beta amyloid plaques, we find the tau inclusions, they fulfill all the criteria for Alzheimer's disease. At the same time, in 9 of the 10 brain we have tested, you see inclusions of TDP43. Of course, somebody would say, you know, TDP43 is the end, TDP43 deposition is the end result of degenerative process which starts with something else. But how can you know about this? I mean, maybe it's the starting point. So maybe, and maybe Alzheimer's disease at the molecular pathophysiology level is not one disease, it's different diseases, one starting from TDP43, the other from APP, from preselin, or from tau, etc.
I'm distressed that you say maybe. Isn't it self-evident?
Yes.
Well, we know, yeah. Go on.
Actually, this is an interesting point because the distribution of TDP pathology and otherwise AD pathology in the Hopkins brain bank is about 45%. You're saying it's 9 out of 10?
Yes.
And so it raises questions about cohort effects. Maybe AD in Crete is different than AD in Baltimore. And I mean that in all seriousness. So it's very interesting to have such a higher rate of positive TDP43 inclusion in your AD pathology page.
That's an excellent point because we proved that our population is extremely homogeneous. So maybe in Boston you have like an admixture of different genetic backgrounds. So you cannot, you have different types of Alzheimer's disease there. So maybe it's the population effect, as you said. Very
where was the last time you saw a pig body in a brain autopsy?
We didn't, in these brains that we have, we didn't specifically search for pig bodies. So we see only the tau and the beta amyloid. So, but maybe it's a good idea to be more specific about this. But
well, I'm just trying to illustrate a point. We routinely stain for them, and we don't ever see them. And yet, in Argentina, it's about 8 or 10% of all autopsies of pig bodies. So there are these regional ancestor-related variations that are important. Anyway, I don't want to monopolize, but I think understanding the functional downstream effect and how they help us differentiate different forms of these degenerative disorders is really where these variants are going to be important.
I just wanted to add that, you know, the TDP43 is really a puzzle worldwide. I mean, we know that in big brain banks, it's quite common in controls, in people without dementia. Very high numbers. I've seen 30%, 40%, 50%. And then I don't know whether the age beyond the ethnicity and the genetic part, I don't know, Yanni, are your autopsy series older people? Because they
are old people and they have advanced dementia because, you know, we have this process of taking informed consent, etc. So
because oftentimes in quite old people, you see it's more common. So that could be another, you know, factor there.
Okay, so the same, that's very correct. The same, of course, stands true for a beta peptide and also for tau inclusions or even alpha-synuclein inclusions. So it's, I think we are far from having a system of characterizing these diseases post-mortemly. And imagine what happens pre-mortemly. We don't have the tools to discriminate. And maybe one of the tools that we could use is the ones that Ioannis presented, the genetic tools. It's not the only one. You need the phenotype, you need different things, but of course it could be a factor. So Kostas says has to see a patient. Thank you very much for the questions and the participation. Is there any other question or comment? Ioannis Tsiverdis, sorry to monopolize this on the molecular field, but you know you are getting into this field slowly. You are a clinician, but since you are working with genetics, I know you like very much this basic research. So, are there any other questions? If not, I don't know if Ioannis Tsiverdis wants to make a final comment. What is his closing remarks on this?
I don't have prepared something for it. I think that still we know a lot of things, but there's a few things in genetics, especially in correlation with the phenotypes that we need to address and to discover. I'm just curious to see where this will go.
So thank you very much. So you have a lot
Thank you very much all.
You have a lot of your work in front of you. Okay, so thank you everybody for being here. See you next time.
Thank
Thank
you.
you,
Bye-bye.
Ioannis.
Bye.
Thank you, Ioannis Square. Okay.